Reproductive system. Uh, Calling it the reproductive system is a little bit of a misnomer, namely because we have at least two systems working here. We have the male reproductive system and we have the female reproductive system. So if you all have been enjoying the sort of lackadaisical nature of AMP2, where it's like, oh, we get one system for every 2 days, I'm sorry. We kind of have at least two chapters of information boiled down into today, but really we kind of add a third, okay? We add a third because we also have to talk about production of gametes, and particularly the process of genetically producing gametes. So we have to talk about this process of meiosis, because what we're really trying to do here with the reproductive system is as simple as we need to get cells from two parents, and we need to have those cells come together to become an embryo. We then have to grow and support the embryo through development, okay? Development is an entirely different chapter, one that we don't have to cover in here. We'll mention it a couple of things. But mostly we need to get to those cells. And the reason that we have to worry about making cells a little differently is because if you just take kind of two random body cells, those two random body cells actually have a pair of every chromosome. So we have 23 pairs, okay? So we have one chromosome, one from mom, and we have one chromosome, one from dad, okay? The reason that's important is because whenever we need to make a new child, that means we have to have one chromosome, one from mom, and one chromosome, one from dad, okay? Which means I can't just pass on my entire chromosome or both of my chromosomes. I have to now separate those chromosomes, okay? So doing ordinary sort of body cell maintenance, we go through a cell division process called mitosis, where you start with your one chromosome from mom, one chromosome, one from dad, and then you just replicate them and you make two from mom and two from dad, and then you just divide those and send in one copy of each off to the daughter cells, and that's at least fairly straightforward. But in our gametes, we have to further subdivide them so that we can only pass on one of each chromosome to the fetus, okay? Which means we have to go through a second set of divisions, okay? That second set of divisions, then we're going to pass on, so in our gametes, in our sperm cell or in our egg cell, we're going to pass on one copy of chromosome 1. We're going to pass on one copy of chromosome 2, and so on and so on and so on, down all the way through our 23 pairs of chromosomes. We're going to call those cells gametes, okay? So our goal then, first and foremost, has to be to produce gametes. Our second goal then is going to be for males to deliver those gametes to the female reproductive system. But for the female reproductive system, it's going to be to receive those gametes, have fertilization occur, and then allow for development of the growing embryo, okay? Now, part of controlling all of this is, of course, going to come from our sex hormones. These are going to be hormones that are going to affect the maturation of the body, but as well as the maturation and development of the sex cells themselves, okay? So whether it's maturation of the organs, development of the organs, and then the organs, or activity of those organs, all of this stuff has to be regulated, and we're gonna do this using our sex hormones. So for males, this is testosterone. In females, it's going to be, the end hormones are going to be your progesterones and estrogens, okay? Now, both sexes, both males and females, are also going to have some accessory reproductive organs. So these are going to be things that are going to carry the gametes away from the gonads, okay? So for males, the gonads are your testes, and females, it's the ovaries. So for males, it's going to be a set of tubes carrying the sperm cells from the testes to the female reproductive system, so carrying them outside the body. So this means we have to deal with the urethra, and the penis and all of these fun things. In the females, we have to go through the fallopian tubes and then the uterus and then through the vaginal openings and all those kinds of fun things, okay? Now, in order to have these two systems come together, we have to have copulation or coitus or intercourse, whatever words you want to use. This is basically to say we have male sex cells passing through the male urethra, and then passing through the vagina and into the uterus, and then finally into the fallopian tubes where fertilization occurs. And then we need to support, protect, and nourish the growing embryo, and all of that is going to happen in the female reproductive tract. Now, uh, the growth and maturation of all of these organs is going to start in what we call puberty. Okay, this is where we get males like getting louder, deeper voices and stuff like that. But this is all going to start in both males and females via gonadotropin releasing hormone, GnRH. So this is going to come from the hypothalamus. When this gets secreted, it's going to then land in the anterior pituitary, which will now release follicle stimulating hormone and luteinizing hormone. These two hormones are going to lead to the production of sex hormones in the gonads. Okay, so remember that the gonads are the testes in males and then the ovaries in females. Follicle stimulating hormone and luteinizing hormone get secreted from the anterior pituitary, which is then going to cause the gonads to secrete their sex hormones. So if it's in a male, the testes will now start producing testosterone. If it's in a female, the ovaries will now start producing progesterone and estrogen. Okay, so whenever that starts to happen, that means we're going to start secreting gametes or producing gametes, and we're going to have maturation of the sex organs, which is to say we have now initiated puberty. Now, importantly, these three hormones, gonadotropin releasing hormone, follicle stimulating hormone, and luteinizing hormone, will now start to be secreted for basically the rest of our lives. Okay, after puberty, we will continue secreting these things. So gametes get produced both by males and females. Females are only going to be secreting a single oocyte every month. So they're going to make one gamete, an oocyte, and they will produce and release a single one of those monthly. They will have several of them in development in various stages of development all the time, but only once a month at ovulation will we release one of these oocytes, usually one. If someone is on something like, say, fertility medications, they can sometimes release more than one in a single ovulation event. Which is why folks that are on fertility medications are more likely to have twins and triplets and stuff like that than is just a normal or like someone that is not undergoing those treatments. Males can produce as many as 100 million in a day. Okay. Now, males are only going to store these for a short period of time. They're only going to be good for a week or more, maybe a month. At which point, if they are not or not used, they simply get reabsorbed, and then you can recycle those parts and pieces to be used later on. Now, helping hold all of these things in place, so helping hold all of these pelvic organs in place, as well as supporting all of these pelvic and reproductive organs, we have a system of muscles that we call the perineum. So we've basically got these two triangles of muscles, both in the female and the male, which basically support and protect all of these reproductive organs. The health of these muscles is actually important to make sure that weird things do not happen to these muscles or to these organs, and make sure that everything works correctly. Now, like I said, we have 23 pairs of chromosomes in the body. We have 22 pairs of what we call autosomes. Those autosomes are basically just normal chromosomes. We're going to number these anywhere from 1 to 22, and we're going to number them based on size. The biggest one is chromosome 1. The smallest one is chromosome 22. Okay. These are going to contain genes that basically control ordinary cell function. Or can just not ordinary cell function, just cell functions, just in general. Okay, these are also going to contain all of the genes that determine kind of physical characteristics, whether it's something like height, hair, eye color, how much skin pigment we have, whether you're like body composition, like all kinds of fun stuff are going to be determined by genes that are going to be found on these autosomes. Okay. Now, importantly, these are paired. So, like I said, you get one copy of chromosome 1 from mom and one copy of chromosome 1 from dad. At which point, those two things are going to have the same genes on them. At which point, we call them homologous. Okay, that they basically are going to contain the same information. Which also means that we can shuffle together the genes on chromosome 1 from mom and chromosome 1 from dad so that we can make a novel combination of those genes. Okay, we call this process crossing over. Now, we also have sex chromosomes. The sex chromosomes is pair 23, where we have two chromosomes, either an X or a Y, or X and a Y, or an X and an X. Okay, these are going to contain genes that are going to determine kind of male and female. And they're also going to contain some genes that code for other functions. There's one gene, say, for in the clotting cascade, which is going to be found on the X chromosome, where if you have or that's found on the X chromosome. Um, the X chromosome is actually quite large and will contain lots of genes for a lot of functions that aren't necessarily even related to sex at all, that just happen to be found on that X chromosome. But because they're found on the X, will be more likely to show issues in those genes if they happen to be found in a male. One of these is for color vision. Okay, so for uh, red-green color blindness, the gene that affects that is going to be found on the X chromosome, which means if you have the faulty version of that gene on your X chromosome, that if it's faulty in a male, you do not have another copy that can mask its presence, which means it's just there in males, which means you don't have your red-green color distinction. So you're red-green colorblind. So it's far, far, far more likely for a male to be red-green colorblind than a female, just simply because the faulty gene is on that X and it can't be masked by the presence of a functioning gene on the other X, like in a female. Because in a female, if you have one good one and one bad one, the good one just does everything that it needs to do, and you'll never know that you have the bad one. In a male, you just have the bad one and it can't be seen by the good one. So now you don't have that function. The same thing goes for hemophilia. So hemophilia is basically a clotting disease. So these people tend to bleed really, really badly. This is actually really common in, say, the British royal family. That one is also an X-linked trait. So it's going to be found on the X chromosome, and thus is going to be way more likely to be seen in males than in females. Now, we call these normal body cells diploid because we have 46 individual chromosomes, so we have 23 pairs. We're trying to produce a haploid cell, which is to say that we have one chromosome 1, one chromosome 2, one chromosome 3. So we have 23 individual chromosomes, one of each pair. Okay. We do this to make sure that the offspring do not receive a paired set of chromosomes. Why don't we want them to have a paired set of chromosomes? Can anyone think of any issues? Keeping in mind that we're all online right now. So can anyone think of any reason why we don't want to have a diploid cell meet a diploid cell? What would happen if we had 23 pairs of chromosomes in one gamete and 23 pairs of chromosomes in another gamete? How many chromosomes would we end up with? You can either type or unmute yourself and say it. What you think? We would just simply have too much information. Like we'd have too many genes, at which point things wouldn't work the way that they're supposed to work. We need to have our genes in the proportions in which we have them. Both the proportions that we have them in is also just the numbers that we have them in. Otherwise, we just simply have too much information and things wouldn't work correctly. And very quickly, we could run into a scenario where the next generation wouldn't have 92 chromosomes. They'd have what is that, 184, and then 368, and then 732, or 736. Like these numbers get really, 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 really fast. So by lowering that information, we can make sure that the offspring do not receive two sets of chromosomes. Instead, they just receive one set of chromosomes, which makes them have the same number as the parent generation. Now we're going to do this by going through what we call meiosis. So in meiosis, we're going to make our 4 daughter cells, not 2 like we did in mitosis. And importantly, the cells are going to now be genetically different from the parent cell because these are now only going to have one copy of everything. So to get that, or to do this, we're going to end up going through what we call crossing over. So our first step here is that we're going to take our chromosomes and we're going to replicate them. Okay. So the same thing is true for mitosis, where our first step is replication. So say we have a maternal chromosome. So we have our maternal chromosome 1, and we have our paternal chromosome 1, and this is in the parent cell. The first thing that we're going to do is we're going to replicate these. So we're now going to get 2 copies of chromosome 1 and we're going to have 2 copies of chromosome 1 from dad. So 2 copies of chromosome 1 from mom, 2 copies of chromosome 1 from dad. Now, at this point, we are now at what we call prophase 1. Now, what will happen here also, though, is that we will have a little bit of exchange occur between 2 of our copies, okay? So we will now have 1 copy of chromosome 1 that's just for mom, and 1 copy of chromosome 1 that's just from dad, but we're also going to exchange some genes in some of these. So we're going to have 1 copy that is going to contain information that is both from dad and from mom, and we're going to have the same thing occur on the other side, where we basically have swapped out a little bit of information from each of these chromosomes. And like I said, we call this event crossing over. Okay? Which means we've shuffled some of these genes around. Okay? And this is going to happen not just on chromosome 1, but this will happen on chromosome 2 and 3 and 4 and on all of the autosomes. Okay? Now, the next thing that will occur is that we will line all these chromosomes up. We will line up all of these chromosomes on the midline of the cell, at which point we call this metaphase. And then we will divide those or split those into opposite ends of the cell. We're going to grab the entire X. Okay, so we're going to grab the pair of chromosomes. So we're going to grab the pair of chromosome 1s. So this is chromosome 1, say 1, 2, 3. We'll grab 1 pair of chromosome 1s and pull them to a pole. 1 pair of chromosomes 2 and pull, and then 1 pair of chromosomes 3s and pull them towards a pole. But importantly here, we're not going to do this in any particular order. We're going to do this randomly. Okay, so we will randomly grab the mostly maternal one or the mostly paternal one and pull it to a pole. The next chromosome will happen randomly. So this way, we don't necessarily say like, oh, you're only grabbing your mostly maternals and pull them towards one end. Instead, it is going to be by chance that this will happen. Okay? So you're only going to have a random chance of pulling each pair of chromosomes towards each pole. And now we get left in what we call telophase 1, where we're going to kind of tie these things off, put a new nuclear envelope around them, and then hold some of these still here. Okay, so this is at the end of meiosis 1. In the female reproductive system, a lot of our cells are just going to be halted at this particular stage. Then we're going to go into meiosis 2. When we go to meiosis 2, we're going to see the same thing happen. Our chromosomes will migrate to the center of the cell, and then we're going to now divide them. Now, we've already done our crossing over, so we don't have to worry about reshuffling these genes or anything like that. But now we have everybody lined up. We're going to line them up in the middle, and now we're going to pull one arm of our X away from each other. Okay, so now we're going to divide those homologs and pull those into opposite poles. So now each daughter cell will have 1 chromosome 1, 1 chromosome 2, 1 chromosome 3. So now we're going to be left with the right amount of DNA in each of the daughter cells. But once again, this will happen randomly. So we're going to basically end up with a random shuffling of all of these genes on all of these chromosomes. So while, yes, you do end up with a maternal chromosome 1 and a paternal chromosome 1, as well as 2 mixed, well, that means you have a 25% chance of passing on your maternal chromosome 1. You have a 25% chance of passing on your paternal chromosome 1. You also have a 50% chance of passing on a mixed chromosome 1 to either or to any of your daughter cells. So 2 of your daughter cells, one of these, or say you have 4 daughter cells. 1 daughter cell will have a maternal chromosome 1, 1 daughter cell will have a paternal chromosome 1, 1 daughter cell, or 2 daughter cells will have mixed chromosome 1s. What this does is this gives us additional shufflings of these genes, okay? And importantly, by having this crossing over event that happened in the first meiosis, we can actually even shuffle within each chromosome 1. So say you have genes that are linked together on chromosome 1. So say it's both a couple of height genes, or say an eye color gene and a hair color gene, okay? So say you have a gene that says, I will likely have brown hair and brown eyes. Well, say that is just from your maternal chromosome 1. You have brown and brown on that chromosome. Well, if we have crossing over occur, it's entirely possible that we can then pass on your mom's brown hair and your dad's green eyes towards the offspring. But then those get meshed with your partner's chromosomes that have also been mix matched, which means it's very easy for us to shuffle all of these genes in, and we end up with essentially an infinite number of variations that we can end up with in the offspring cells. So this way, you're never going to have an offspring that's going to be exactly like you or exactly like your parents. And that's going to be because we're shuffling your parents' genes around. So the parents' genes around that made up you, that you even reshuffle those to make it where you're passing on something that can't be exactly like you. And then you're mixing them with the genes of someone else, which are also not like you. So you're going to end up with a novel combination basically every single time. Now, it's important here that we do this process correctly. Okay. It is very important that we divide the chromosomes correctly so that we have the daughter cells getting one copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, so that we don't have a mismatch of numbers, because we want to have an offspring that has exactly 46. If they have 47, we call this trisomy, because that means they're going to get an extra copy of one of those chromosomes. If that happens on anything that is not chromosome 21, it will be lethal to the embryo. Okay, most, but not all, but almost all trisomies or monosomies, that's to say that we end up with one fewer copy of one of the chromosomes. So say we don't end up with 2 copies of chromosome 21, that we end up with instead 1 copy of chromosome 21. Anytime we end up with too many copies or too few copies, that is almost always going to be lethal to the embryo. One exception of this is trisomy 21. Okay, so remember that we number from largest to smallest. So trisomy 21 is going to basically mean it's the second smallest chromosome that we have. If you get one extra copy of the second smallest chromosome that we have, you end up with what we call Down syndrome or trisomy 21. This leads to lots of kind of fallout, whether it's some intellectual issues that can occur, the slanting eyes, heart defects, muscle tone, all of these kinds of things, all of these issues that we tend to relate with Down syndrome are all because they have one extra copy of the smallest chromosome. One extra copy of, say, chromosome 1, those embryos do not make it past a couple of cell divisions, at which point they will basically like stop cell dividing and go through programmed cell death just on their own, just simply because they can't continue developing because of all the issues that arise because of having too many of those chromosomes. Now, the other chromosomes that we can, or the only other chromosome, or yeah, the only other chromosome that we can end up with issues or end up having more than one of or too few of is going to be the sex chromosomes. It is actually quite common for people to have 3 sex chromosomes, either XXX or be XXY on their sex chromosomes, and they can live to sexual maturity and they can live kind of normal, healthy lives. Or it's also possible for someone to be what we call X naught, which is to say they got 1 X chromosome from likely mom and then nothing from dad. So they will end up with kind of having no secondary sex chromosome. But once again, they can live to like normal, healthy, or like long-lived lives and stuff like that. But those will also all lead to kind of their own sort of indistinctions when it comes to some characteristics and traits and things like that. But typically non-disjunction, that's to say having too many copies or too few copies of chromosome, will be lethal, and they'll just be lethal at various times of like through development. All right, so does anyone have any questions as far as like genetic stuff is concerned? No? All right, then we'll just keep moving on. All right, so female reproductive system. What are we trying to do with the female reproductive system? Well, female reproductive system, we have a few things going on here, okay? For the female reproductive system, we of course have to start with the actual sex cell producing organ. That's going to be our ovaries. So here we have our gonads here. This is also going to be the organ that is going to be making our sex hormones for the most part. And that's going to be coming from the ovaries here. Now, the ovulated oocyte will then be collected by this little hand-shaped structure that we call the fimbriae. These are basically at the opening of the uterine tube. And then those will lead down to the uterine tube, or what we call the fallopian tube. There's one of these on both the right and the left. Those eggs will then pass down through those uterine tubes and then finally down to the uterus. That's the structure here. Now, this is a big muscular structure containing 3 layers of tissue, where we have the perimetrium on the outside, we have the myometrium in the middle, myo meaning muscle, so we have the muscular portion of the uterus, and then finally we have the endometrium, which is going to be the epithelial lining on the inside, okay? Now, during menstruation, this is actually the process of shedding the inner endometrial wall. And that's going to happen by contraction of the muscles in the uterus. So the muscular lining of the uterus will then contract and squeeze and contract and squeeze and contract and squeeze so that we can squeeze out all of that endometrial lining so that we can then pass that out through the vagina to make room for another endometrial lining to grow over the course of the next month. Maybe make this, come on, pointer, where'd you go? All right, now, that is then going to lead us down to this little narrow region right here, which we call the cervix. Okay, so the cervix is basically going to be an extension of the uterus into the vagina or the birth canal. It's got a very small opening right here, which is going to prevent things from passing into or out of the uterus. Now, after that, we're going to then lead into the birth canal, which is here. So this is the vagina here. And then that will then pass out through the birth canal. And then finally, we're going to get to the external genitalia, which is out here as the labium majus and the labium minus out here. These are basically some flaps of tissue that are going to protect the openings of both the vaginal opening and the opening for the urethra. Okay, so we're going to protect those with a couple of little bits of tissue to make sure that foreign material can't get in there. Just kind of protect all that. Okay. Now, we can also see a little bit of erectile tissue here. I say it's erectile tissue because we can fill this with blood. Okay, so it's got a bunch of little, it basically looks like a little sponge. So if you were to zoom in on that, it would basically look like a little sponge, which means we can fill it with a bunch of blood. And when it gets filled with blood, it will basically make it become firm. Now, this is only from this medial view. The clitoris here will actually extend around the opening of the urethra and of the vaginal openings all the way down here. Okay, or you all can't see where my pointer is. All the way out here, wrapping around the opening of the vagina. So whenever that fills with blood during kind of sexual intercourse, it will actually allow for some of the sensation of intercourse will actually be from pressing up against the walls or engorged walls of the clitoris as it's wrapping around here, wrapping around the vagina. Now, if we look from an anterior view, we can see that the pubic bone is covered with a little bit of fat here that we call the mons pubis. It basically just protects the really sharp anterior portion of the pubic bone from the outside, okay? So here we have the urinary bladder, and then finally we have the uterus here. We can also see the ovaries back here. The uterus is going to be protected, or at least held in place by a couple of structures, namely our round ligaments, holding them in place anteriorly, and then the broad ligament holding them in place kind of laterally. The ovaries are then held in place by ovarian ligaments up here, okay? Now, you'll also notice that the uterus and the bladder are very, very close together, so much so that movement in the uterus can oftentimes cause motion in the urinary bladder itself. And if you put a little bit of pressure on the urinary bladder by like pushing on it a little bit, like say from the uterus, that will cause additional pressure on the internal urethral sphincter, which remember is down here, which basically is part of where we're going to get the sensation that we need to go to the restroom. Notice that we have uterus basically sitting on top of bladder here, which means during menstruation, when these muscles are cramping and stuff like that, that can put additional pressure on the bladder, make it seem like you need to go to the bathroom more. During pregnancy, when we're taking this spot and filling it with a baby, it's going to be putting that much more additional pressure on top of the bladder, which means folks that are pregnant are going to feel like they need to go to the bathroom basically all the time because the bladder can't grow all that big and because any movement inside the uterus here is going to cause additional pressure on that internal urethral sphincter, which basically is going to send the signal that says, hey, I need to go to the bathroom. So they're going to receive that signal a lot more than someone that is not pregnant. So here we can see some of our uterine ligaments holding everything in place. We have the ovarian ligament holding the ovary to the uterus here. We have the broad ligament helping hold everything kind of in place laterally. And then we have the uterine ligament holding things in place anteriorly. We can also see our cervix and the opening there called the os, or the external ox. And then we have the vaginal opening down here. Now, if we were to take a look inside the ovary, at any given time, we're going to see several groups of cells in this kind of progression, okay? We're going to start here with cells that are what we call primordial follicles, okay? These cells are all going to be present at birth, and they're just going to be kind of halted in that first meiotic prophase, okay? That's to say they have had their DNA duplicated, where we have chromosomes that look like they're in little X's, and that's about it. We've got duplicated DNA, and they're basically waiting to go to the next stage, okay? Remember where we're moving a whole entire X, or a whole entire like paired chromosome 1 into the offspring cells, okay? Then we move into our primary follicles. That's going to be these cells here. And then finally into secondary follicles and antral follicles, where we get this kind of fluid-filled tissue here called antrum around our corona radiata. And then finally, we move to a mature follicle, at which point this oocyte in here is ready to go and is ready to receive a sperm cell, at which point it will finalize going through meiosis 2. But that will only happen after it has been ovulated. So this cell right here is still waiting in a step of meiosis 2. It has not finished meiosis 2 just yet. But then we will ovulate this cell. So we'll ovulate our secondary oocyte right here, at which point this big fluid-filled structure back here will now become a new gland. That new gland is what we call the corpus luteum, okay? So luteum, as in luteinizing hormone. Luteinizing hormone will actually cause us to go from mature follicle and push us in the direction of becoming the corpus luteum. So we're going to go from mature follicle to developing corpus luteum, and finally to produce our corpus luteum because of luteinizing hormone. Okay, that lutea part just basically means yellow. So we have this kind of yellowish structure right here. So corpus luteum, this one's going to produce progesterone and estrogen. So it's going to be a short-lived gland. It's going to produce progesterone and estrogen for about 2 weeks, at which point it will degrade and it will become what we call the corpus albicans. Albicans because it's going to be white. This is basically going to be now a little bit of a scar tissue that will eventually get degraded and just kind of reabsorbed over time. So those primary follicles, so that second stage, these are going to start secreting estrogen. Okay, so once we become those primary follicles, we will now start to secrete estrogen. That is going to cause us to increase the buildup of the uterine lining, so of the endometrial lining of the uterus, okay? So that's what's happening here in our primary oocytes. Then those will become secondary follicles. These secondary follicles will grow many different layers of those granulosal cells, and then we're going to get to thecal cells. The thecal cells out here are going to take the androgens that were getting produced by our granulosal cells here and start to convert those androgens specifically into estrogens. Not just androgens, but into estrogen specifically. So that's going to kind of happen out here with our thecal cells. One way of kind of thinking about thecal cells is, to me, I kind of see thecal cells as essentially the shell of our egg here. That's kind of just the way I imagine it and kind of the way I remember them. It's not correct necessarily. That's just one way of kind of thinking about them. All right, then we go to our antral follicle, where we're going to have this kind of fluid-filled antrum, okay? When this happens, we're going to get this little corona, or this little crown called our corona radiata around our primary oocyte. So these little cells here we call our corona radiata. Here we have the antrum, that's basically kind of a fluid-filled cavity at this point. Once again, we're still trying to grow the cell, get it larger, and basically prepare it to become an embryo if it needs to do so. Then finally, we go from antral follicle into mature follicle. Our mature follicle is going to be this big structure right here. Or this whole thing is our mature follicle, where we have this big, heavy antrum. We have a big corona radiata. And then we still have our secondary oocyte right here in the middle, as well as that little thin layer of our zona pellucida outside of it. Okay. At this point, we have completed meiosis 1. Okay. We are now held in meiosis 2, particularly in metaphase. So right now in this stage, this cell has all of its chromosomes, duplicated chromosomes, and they're held in place in meiosis 2. So they're lined up in the middle of the cell, which means they're simply waiting to pull apart. Okay, so we're basically waiting to make the final decision on what the genetic information for the daughter cells is going to be. Now, we're going to produce one of these a month under ordinary circumstances. Some people have twins, like fraternal twins, that are very common in their kind of family tree, at which point those people are going to be more likely to produce more than one mature follicle every month. So they may release 2 mature follicles a month. So they might release one for both the left ovary and the right ovary every month, whereas most people should or will only release 1. But that's kind of how we lead to those fraternal twins in those situations, is that we've released 1 from each ovary. Now, after ovulation, so we release one of those mature oocytes every month, then the antrum and the stuff that's left will then become the corpus luteum, at which point we will secrete massive amounts of progesterone and estrogen. By doing this, we're going to stimulate the buildup of the uterine lining. So we're going to build and tell the uterine lining to get even thicker and thicker and thicker, so that it has more space to, or is going to be a more hospitable environment for a fertilized zygote to land, or a kind of proto-embryo to land. And then at which point it will get all the nutrients and stuff it needs from the uterine lining. But once again, if we do not have fertilization of the zygote or fertilization of the egg, we will then take that corpus luteum and it will become the corpus albicans, which is basically some connective tissue. And then eventually those structures will be completely reabsorbed. It just takes a little bit of time. I love this figure. This figure makes it really easy to understand a bunch of the different changes that are happening, both going from our primordial follicle all the way up to our mature follicle up here, where we can see some of these changes, but we can also see the hormones that are causing some of these things to occur. So follicle stimulating hormone is going to be at reasonably high levels in the first 7 or 8 days, or 10 days even, of our monthly cycle. So of a normal 28-day cycle, follicle stimulating hormone is going to be fairly high for the first 9. Okay. Follicle stimulating hormone is telling us or causing us to mature these follicles. Okay. So to tell us to go from our antral follicle to a mature follicle. One type of fertility medication would be to stimulate or simulate follicle stimulating hormone, at which point you would produce not 1 mature follicle every month, but you might produce 7 or 8 mature follicles in a month. Okay. Then the whole time this process occurring, we're going to have a slow increase in luteinizing hormone, which is kind of pushing us towards becoming the corpus luteum, remember? And then we're going to see a big spike of luteinizing hormone right here on day 13. By doing that, we're also going to have a big spike on day 13 of estrogen. That's going to be this little graph down here. When luteinizing hormone and estrogen spike, those are then going to cause the release of the secondary oocyte. By releasing that secondary oocyte, we call this process ovulation. And this usually occurs on day 14. Okay, so day 14, we release that cell into the uterine tube so that it can be fertilized, which will happen in the uterine tube. So fertilization will happen in those uterine or fallopian tubes. But that process does not get started until we release or ovulate, which will be because of the effects of a spike of both estrogen and a spike of luteinizing hormone, causing that mature follicle to turn into the corpus luteum. When that happens and we start to produce that corpus luteum, we're going to see a big spike in progesterone secretions and then another spike in estrogen secretions. As that happens, we're going to quickly increase how much of the uterine lining we are producing. At which point, we now have a uterine lining that is now conducive to a growing embryo implanting. So it will bind, it will stick to this big growing structure up here, which will also have lots and lots of blood vessels growing in it. And then those blood vessels will then grow and connect to the growing embryo, at which point we will not shed it because we're going to secrete a hormone called human chorionic gonadotropin, which basically halts us right here. So we're going to continue producing estrogen, progesterone, so that we don't get rid of it. If we do not have an embryo bind here, though, these levels will fall off as our corpus luteum degrades. When that happens, we will then shed that endometrial lining and we will start this process all over again. That's why we call it the menstrual cycle. Now, the menstrual cycle is also going to stop around between the ages of 45 and 55. We call this menopause. It usually starts by being somewhat irregular, and then finally it will just stop at some point between 45 and 55. At which point, or at which point you are no longer going to be fertile. Because you're no longer going through this process, you're no longer going to be releasing a follicle every month, which means sperm cells will not have an oocyte to grab onto, at which point you cannot have a fertilization event. This also means, though, that you will stop shedding the uterine lining over that time. Okay. Progesterone will now be the primary hormone that's getting secreted. Estrogen will stop getting secreted in as high of levels. Okay. Now, there are some hormone levels that have to be regulated in here. And that's kind of what these graphs are showing you, is kind of high levels or low levels of these things leading to either or leading to negative feedback. And you can kind of look at that a little bit. I'm trying to move a little bit so we can get back to some anatomy things. All right. So once again, we have the fimbriae down here, which are going to be the openings of our uterine tubes. And we're going to call this first little bit the infundibulum, or so the fingers would be the fimbriae, the hand part would be the infundibulum, and then that will lead to the actual uterine tube. Now, sometimes weird things happen. Like I said, that we have fertilization will occur here in the uterine tube. After that happens, we're going to start to see some cell divisions of those cells kind of occurring. And for the first several, they're going to stay the same size overall. We're just going to kind of subdivide the volume of those cells, which is why it's so important for that cell to mature correctly, is so that those first several cell divisions can happen right. And then that kind of dividing cell will land in the endometrial lining, at which point it will start to grow and develop. However, sometimes things happen strangely, and that fertilized oocyte implants outside of the endometrium. So it will land sometimes in these tubes, and we would call this a tubal pregnancy, or ectopic basically just means anywhere outside the endometrium. But primarily, this would be a tubal pregnancy more often than not, where it tries to implant in the uterine tube, at which point the uterine tubes are not able to grow and expand the same way that the uterus does. So there's really no room for it to grow, at which point the embryo is only going to be viable for a few weeks of development before it will start to cause kind of the rupture of the uterine wall, or and if that wall ruptures, that means you're going to have hemorrhage and potential loss of life, not just of the embryo, but also of the mother at this point. So these tubal pregnancies can be quite dangerous and can lead to kind of lifelong issues with fertility and stuff, because basically that side you lose access to because you essentially have to, that basically messes up its ability to work anymore in some cases. So those can be somewhat scary, and they absolutely can be life-threatening. So once again, we have our endometrium here. We have the myometrium, which is this muscular portion, and then finally the perimetrium on the outside. And then we have the external os down here, which is basically going to be the opening of the uterus into the vaginal canal down here. Okay. Now, during childbirth or during labor, so before kind of the start of childbirth, when people are looking for dilation, they're saying, okay, I'm dilated to a 6 or I'm dilated to a 4. What they're actually measuring is this particular opening right here, not the opening of the vaginal wall itself, but actually of the external os, so the opening of the cervix here. Usually childbirth will not happen until this is at an 8 or a 10. So at 8 to 10 centimeters, which is roughly the size of my finger here, when ordinarily it is like the size of the end of a pencil or like the lead of a pencil, like maybe a millimeter in size. So it has to get a lot larger. So going into like the centimeters, which is just crazy to me. But if you've ever seen a baby, that means you literally have to make that opening the size of a baby's head, which is one of the many reasons why I'm glad I'm a guy. I cannot imagine having to, one, carry a baby, much less two, having to push one out through here. Like uh, our first child, so like our son weighed 9 pounds and 15 ounces. He also pooped like literally the second he was born. Uh, so there was like an ounce there. So he was a 10-pound baby and he had to go through that opening. So I just cannot. My wife is a superhero. Like, yeah, nope. Like our second kid was born a week premature. So like my son was born 1 week late. So because of that and because of his size, we actually induced a week early for our daughter, and she still weighed 9 pounds or 8 pounds, 15 ounces. So she would have been ginormous had we let her go much longer. My wife, like I said, is a superhero and a saint. Also, with our daughter, she actually broke a toe one day walking into work uh, and worked the entire shift as though nothing had happened. Didn't realize that it was actually broken until like later that night when she took her shoes off and her toe was just black. Great. Or not, well, not black, but like kind of dark blue purple. So here's the endometrial lining. We have, it's the functional layer that we're shedding, not the basal layer. But this layer out here basically gets soft, really, really vascular so that we can allow for the embryo to implant. The vaginal wall is going to be composed of non-keratinized stratified squamous epithelium, basically a big mucosal layer. Why? Because it's going to be subject to abrasion both from childbirth, intercourse, but also because it's open to the outside environment. We want to make sure that we have a nice thick layer of cells between potential outside environment and our internal environment, so our muscularis. So we do have some non-keratinized stratified squamous epithelium here to help deal with all of that. Talked about that. Oh, here's a picture of the internal structures of the clitoris here. So the clitoris basically splits into these kind of Y-shaped structures here, where they will go around the vaginal opening down here. Just those are all internal structures, so you just can't see them. All right, then we get to the mammary glands. So the mammary glands are important because these are going to be, of course, producing breast milk after childbirth. Okay, so here we have a bunch of tissue. So we have a bunch of mammary tissue. These are basically just glands. So these are all going to be held in place with some ligaments. And we have a bunch of glands connected to a bunch of ducts, which will then bring those lactiferous ducts and push them out through the nipple. At which point, when these things start to produce a bunch of milk, that milk gets pushed out into these lactiferous ducts, at which point they will then get expressed out through holes in the front of the nipple here. Okay. Now, unfortunately, because it is milk and milk is fatty, sometimes if the milk is too fatty, it can actually lead to clogs in these ducts. Another thing that can lead to clogs in these, or just basically fat clogs in these ducts, just like imagine like butter moving through these, basically get it a little bit too thick, and then you end up with a little bit of a clog, but you push with some pressure and you can kind of force those clogs out. Other times you can have clogs get caused by infections, because milk is the perfect thing for a growing embryo because it contains high amounts of sugar, fat, and protein, all the stuff that makes milk awesome. For growth for a human also means it makes milk awesome for growing bacteria and fungi. So sometimes you can have fungal or bacterial infections of these ducts also lead to clogs. And when that happens, it can lead to a clog and you end up with a bunch of buildup of milk production back here, which can swell and kind of you'll end up with inflammation and redness and high fevers and stuff like that due to clogs of these ducts and lots of pain. We call this mastitis, which is very much no fun for the patient that is undergoing that. Now, if you've ever heard of someone having a mastectomy because they have a history of breast cancer or something like that, or they have cancer, what will happen is the surgeon will go in and remove all of this mammary tissue, so all of the glandular tissue. Okay. Unfortunately, though, some of that glandular tissue is actually still connected to the nipple up here. So if you leave the nipples in place, you actually do risk leaving some amount of mammary tissue up here, which can actually still start having cancer. So even if you have had all of this big stuff back here removed, you can still end up with cancer here. So oftentimes, whenever a mastectomy happens, they will come in and they'll literally take out all of the nipple structures as well, just to make sure they can get all of that tissue out. Now, oftentimes they would basically just fill this little pocket where all of this mammary tissue was, and they'll fill that with some silicone, like a silicone implant, so that someone will still have something that will look like the breast there by basically just filling in this space. Uh, Other times, what they can do now in some fancy surgeries is they'll actually go down to the abdomen and they'll actually grab some fat from the abdomen as well as some of the blood vessels that feed that fat. And then they'll actually bring that whole structure up here to fill in all of this space. And then they'll basically tie in that fat with the mammary arteries and veins, so that you have blood flow in and out of that donor tissue. That way, you still have something that is going to look like a breast, but without having any of the mammary tissue up there. So that's one of the other ways that they can do that. But when a baby suckles, that it's actually going to cause a release of oxytocin from the hypothalamus. And when that happens, that will actually cause the muscles associated with these glands up here to kind of squeeze, and that will actually cause ejection of the milk out through the nipple there. And all because of the suckle of the baby. We'll actually be kind of suckling in a particular pattern on the nipple, which will cause that, what they call a letdown or a release of the milk from there. There are some different methods of contraception that they're going to list here and kind of show you their rates of failure and things like that. Whether it's barrier methods like condoms or whether it's from intrauterine devices or oral contraceptives, all of these things and kind of telling you how those work a little bit. There's lots of information out there. All right. In the male system, things are a little bit different. In the male system, what we're trying to do is not grow a single oocyte every month and then deal with maturing that oocyte and then releasing it to a particular place for fertilization and then having to house the growing embryo. Instead, it's going to be production, secretion, and delivery of gametes. Okay. So because the vagina is interior, the male penis has to then be found exterior. Okay. Now, in addition to the penis being exterior because it has to deliver the cells outside of the body, we also have the testes being found exterior of the body because sperm production has to happen a couple degrees below the temperature of the body. Okay. So normal body temp is like 97 degrees or 97.5 degrees. Sperm production needs to happen around kind of 93 to 95-ish degrees. Okay. So because of that, the testes are going to be found outside the body. They're going to be found like a little bag here that we call the scrotum. That's going to contain a couple of layers of muscle tissue to help position the testes in various distances away from the body so that we can help kind of regulate the temperature of those testes. Now, the testes themselves and testosterone are going to be produced here in the testes. Sperm will then be stored in this kind of big structure here on the testicle or on the testes, and then we're going to call this the epididymis. Okay, so this is our epididymis. And then during ejaculation, the sperm will be pushed out through the ductus deferens or vas deferens out here behind our urinary bladder, at which point it will come into contact with the seminal vesicles here. Okay, so vas deferens or ductus deferens, and then finally seminal vesicle. And then finally, that will then pass into the prostate gland. That's this kind of structure right here. It will then go into or past the bulbourethral glands, and then finally into the spongy urethra of the penis, and then finally out through the glans penis. Okay, that's kind of our pathway of production and out. So we have this big kind of circular pathway out. Now, the penis is going to be composed of two main layers of erectile tissue. We have the spongus or cavernosus spongiosum, which is around the urethra. Thus, we have the spongy urethra, but we also have the corpus cavernosum, which is going to be this region up here. This is basically all spongy tissue. So it's a whole bunch of kind of airy tissue that we can take all these little thin bits of tissue and fill them with blood, at which point we can make the penis be held erect and firm. Now, that is in humans. Some mammals will actually have penises that will actually have a bone in them. We call that bone a baculum. That's going to be found in some stuff like dogs and in stuff like raccoons. They will still have the erectile tissue, but there will actually be a bone in there that will help support that, but not in males or not in humans. Humans do not have a penis bone. Okay. Instead, it's basically just going to be blood that will help hold all of that firmness and stuff there. Now, I'm trying to think what else we need to talk about. All right. So when we look anterior and we kind of look at the testes down here, we can see two layers of muscle. The first layer of muscle, just deep to the actual skin of the scrotum, is going to be called the dartos. Okay. So the dartos muscle is basically going to hold the scrotum together. So when the dartos muscle contracts, it's going to basically take the right and left testes and pull them closer together, once again, to help retain heat. Okay. The same time that happens, we have some muscles here called the cremaster muscle, which are going to lead to either the right or left testes and kind of wrap around the entire spermatic cord. When the cremaster muscle contracts, it's going to pull the testes up. Okay. So the dartos muscle will pull them side to side and pull them in. The cremaster muscle will pull them up. At which point, when it is cold, these muscles will contract and pull the testes up and in and closer to the body trunk, so we can keep things nice and warm. So that way we are at least warming up what's happening here so that we can continue doing sperm production as normal. When things get hot, those muscles will relax and allow things to move away from the body so that we can try and cool them off that way. At the same time this is happening, we have this little network of veins right here wrapping around the testicular artery that's kind of moving its way down towards the testes. So because this is an artery, so it's carrying blood away from the heart, so it's carrying blood towards the testes. Well, the veins are carrying blood back in. Well, if we have these testes away from the body so that they can get further away from the body trunk so that they can cool, that means venous blood is cooler than arterial blood down here. So that venous blood is going to be nice and cool. So we have cool blood moving up and we have warm blood moving down. Well, what does that do? Well, that means we're going to have that cool blood acting as a heat sink for the blood that's on the way out or on the way down. So the blood that's coming down towards the testes will then pass off its heat towards the blood that's on the way back up into the body. So we're going to warm the venous blood in the pampiniform plexus while at the same time cooling arterial blood that's on their way down to the testes. So we have three ways of controlling temperature in the testes. We have the dartos muscle, the cremaster muscle, and we have the pampiniform plexus, which is a vein. Now, this all brings us down here to the testes. So we have our actual testis down here, where we have basically these lobes inside the testes. And these are actually where we're going to be filling these with what we call seminiferous tubules. So all these little tubes here are going to be called seminiferous tubules. And these are actually going to be where we make the sperm cells. Okay. Now, those sperm cells, after they get produced, will then migrate over here into the epididymis, where they're going to then be stored. And if they are not used, they will simply then be reabsorbed. If they are used, they will then pass through what we call the ductus deferens or the vas deferens. If you've ever heard of someone having a vasectomy, ectomy just means to cut. They're going to basically cut this tube, and oftentimes they'll tie it in a little knot and then burn one of the ends or both ends of it. So that way we can't reconnect them via healing. When you do that, what happens is you will produce sperm, but those sperm will have nowhere to go. So during ejaculation, no sperm will actually be ejected out the penis, simply because there's no way for us to move through this particular little tube. Okay. Now, if you were to take a look inside of one of those seminiferous tubules, this is what you would see, where you have the lumen here and you have the outside out here. Okay. Now, out here on this outermost layer, we're going to have what we call spermatogonia. These are going to be the cells that will produce the sperm. So these cells will basically then undergo meiosis 1 and meiosis 2. As you move from here to here, these cells are basically in various stages of meiosis 1 and meiosis 2, where when you get to the spermatids here, or the sperm cells here, these are completely through both meiosis 1 and meiosis 2. So these have completely divided all of their genetic material. Now, we can also see a couple of other cells here. Namely, we have our interstitial cells, which are important because these are actually secreting testosterone. And then we've also got our sustentacular cells here. Sustentacular cells are basically monitoring what is happening inside this tube. If you fill this lumen with a bunch of sperm cells, basically to say, hey, I have produced enough sperm cells to fill this area, these sustentacular cells here will then secrete a chemical called inhibin. Inhibin is going to inhibit the production of additional sperm cells. So if you have produced enough sperm cells that you have enough basically in storage, you will produce inhibin so that you do not continue to secrete follicle stimulating and luteinizing hormone, so that you can bring those levels down, because you don't need to you don't need to make more because you don't have any room for them. That's basically going to happen because the sperm cells are getting produced, but they're not getting used, okay? So the way we regulate here is hypothalamus secretes gonadotropin releasing hormone, like we talked about before. That stimulates follicle stimulating hormone and luteinizing hormone, which in turn calls us to make both spermatogenesis, so making sperm cells, while also at the same time making testosterone. However, that testosterone then tells us to make additional sperm cells. However, if we have high levels of testosterone or high levels of sperm cells, we will then act as negative feedback so that we can bring these numbers back down. So high levels of sperm cells or high levels of testosterone will basically tell us to bring those levels back down. And if this happens because of, say, exogenous testosterone, so someone is getting testosterone injections for athletic performance or something like that, what you'll see is that because testosterone levels are so high, you're going to get this big negative feedback loop where it's going to continue to say, hey, we need to produce less testosterone and we need to produce less sperm cells. And then you keep adding new testosterone in with a needle. What you're going to see is that you'll actually see the testes size shrink because they no longer are needed to get testosterone levels up. So sperm levels will go down, and testes size will actually go down as well. So that's why shrinking testes are actually a side effect of things like steroid use. So we can see this kind of feedback loop here. So make sure y'all spend a little bit of time with that on both the male and female side. Trying to think of any different anatomy we need to look at. Oh, so each of the accessory organs, so both of the seminal vesicles, prostate gland, and bulbar urethral glands, each of these is going to secrete its own component of seminal fluid, okay? So it's not just sperm that's going to be found in seminal fluid, okay, or in semen. It's also going to be all of these outputs here, okay? The seminal vesicles are the first one. These are going to provide the bulk of the seminal fluid. These are going to produce both an alkaline fluid as well as fructose and prostaglandins, okay? So we're going to secrete an alkaline fluid that is high in sugar and high in prostaglandins. The sugar is going to nourish the sperm. The alkaline fluid is going to fight the acidic nature of the vagina. So that way the acidic vagina does not kill off all of the sperm cells. So we have to basically balance out that pH to keep the sperm alive. The fructose will nourish the sperm because the sperm don't really have a whole bunch of cytoplasm to store their own. So you basically have to provide them with some fructose so that they can continue to swim. And then finally, the prostaglandins. The prostaglandins are going to tell that external os of the cervix, which remember is very, very small, to open a little bit so that more of the sperm cells can make their way through, because they have to swim not just through the cervix and through the uterus, but they also have to swim up into the fallopian tubes to try and meet the cells, okay? The prostate glands are going to secrete some mucus, okay? So these are going to secrete some mucin, and these are also going to secrete some citric acid and seminal plasmin, and then finally prostate-specific antigen. Okay, so this is basically going to be a milky fluid that is high in citric acid, which is going to be a nutrient for sperm health. So making sure that the sperm can continue to have high levels of energy and continue basically to stay healthy, as well as an antibiotic. Okay, this seminal plasmin is an antibiotic that makes sure that you don't get a urinary tract infection. Okay, this is one of the reasons why, or that males will also tend to have fewer urinary tract infections, is because of the presence of this seminal plasmin that kind of protects those walls. Okay, and then finally we have prostate-specific antigen. Prostate-specific antigen is basically there to help take all of the sperm, which is very, 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 very thick, like there's really no liquid component in the ductus deferens, and basically allow that stuff to get more liquidy. So it goes from being like jello to being something that is a lot more liquid, to allow the sperm to kind of spread out into that liquid. And that's where we get that prostate-specific antigen. So it's very important there. And then finally, we get to the bulbourethral glands. Bulbourethral glands are those tiny little balls right there that are roughly in line with the pubic bone. These are very important because these are actually going to secrete some mucus that is going to coat and lubricate the urethra during intercourse. Okay, so right before all of the sperm and seminal fluid and stuff like that is going to follow during ejaculation, there will be a little bit of secretion secreted from the bulbourethral glands that makes sure that the urethra is coated with a mucus so that the sperm does not come into contact with, say, leftover urine inside the urethra. So we protect the sperm that way. When I say sperm, we are releasing anywhere from 2 to 500 million spermatozoa in a single intercourse event. That is a lot. One cell gets released from an ovary every month. 2 to 5, or anywhere from 200 to 500 million spermatozoa in a single intercourse event. That is kind of the difference in the number of cells here, which is just kind of crazy to me. And it takes about 2 weeks to go from a seminal tubule inside the testes to being part of ejaculate. All right, we've talked about the penis a little bit here. So we've got a couple of pieces. We basically have the root of the penis down here, which is going to be some stuff that's found inside, but this is still erectile tissue. We have our corpus cavernosum being up here. It has a blood supply, so a central artery on either side. Blood comes in here and basically just gets trapped inside these sponges, and that's where you're going to get the kind of turgor or the pressure. So the hydrostatic pressure that will build up inside the erect penis, and the same thing will happen down here in the corpus spongiosum. But you don't have the main central arteries like you do down up here. But this is actually where their urethra is going to be. If you've ever heard of someone having a broken penis, what has happened is basically you have 2 water balloons here that are filled with blood. And just like a water balloon, when you throw it on the floor, you can cause the walls of that to rupture. And that can actually happen with massive amounts of pressure here on the walls of the corpus cavernosum. That's what's happened when someone has had a broken penis, because there's no bone to break, but it basically is going to be like breaking the walls of a water balloon, at which point it's not water that's going everywhere, but instead blood, which is moderately terrifying. Now, we did mention before that we have genetic sex, where we have an XX for someone that is genetically female, or XY for genetically male. But please keep in mind that there are big differences, or it's also possible for someone to be XXX or XXY, as well as X naught. So there's lots of possible variations here, and those variations are actually surprisingly common. Like 1 in every couple thousand, 1 every 1,000, something like that will be one of those 3 other variations. Now, during development, early on, everything will start the same, but fairly early on in development, like say between weeks 5 and 6, or even weeks kind of 10 to 12, you will have been pushed into either the male direction or the female direction based off of that genetic signal and the hormones that those things provide. Now, someone that is XXX or XXY or X naught will have varying levels of these hormones. So will sometimes be somewhere in between these 2 kind of progression extremes, at which point the fetus will not develop what would be considered normal genitalia on one of these. So usually hormone replacements can be used to kind of push the embryo in one of these two directions based off of that. So these are kind of your intersex states here. So that's basically all of the anatomy and physiology for this chapter, and I guess also this course. So first off, let me just thank y'all. Y'all have been a great class. I have enjoyed teaching y'all this semester. Hopefully you've learned something that you find useful. But I also have all the faith in y'all in the world that y'all will do just fine on the coming exams and stuff. So with that, anyone have any questions for me?
No.
I think I heard someone try and say something. Maybe it was just saying no. Well, I guess in that case, I wish y'all all the luck in the world, both on your finals for this class and all of your other future things. So just let me know if you need anything.